Hello, and welcome to Fluidigm's FAS Friday session for infectious diseases and vaccines. My name is Daniel Frederick, and I'm a mass cytometry FAS based in Boston, and I support Rhode Island, Massachusetts, New Hampshire, Vermont, and Maine. I will be taking your questions after the presentation. Please feel free to enter questions in the question box at any time during this presentation. I will answer them as many questions as possible after the presentation concludes. First, I'd like to discuss how the use of mass cytometry and imaging mass cytometry enables the studies of infectious diseases and vaccines. So first, we will look at, in the first section, with looking at mass cytometry and how it's applied to infectious disease research. So pathogens that breach our frontline barriers are typically met with an aggressive immune response. Slide here uses COVID-19 as a model to show the immune response to viral infection and the complexity of disease progression. The slide shows three qualitative outcomes: asymptomatic infection, mild infection, and severe infection. While there are still many factors to be investigated in relation to COVID-19, the current understanding is that a timely immune response with the hallmarks of early involvement by the innate immune system, along with a coordinated T-cell response, can effectively control SARS-CoV-2. However, in severe infection, lymphopenia is observed along with an increased involvement of macrophages and infiltrating neutrophils. This type of immune response results in a cytokine storm and a type of viral sepsis. Being able to understand the differences in the response of such a disease requires a broad view and deep inquiry. The process of clearing infection typically involves a biphasic immune response, activation of the innate immune system, and then the recruitment of adaptive immune cells and antibody production. In many cases, the innate immune system can single-handedly clear pathogens before an infection ever occurs. The picture is more complicated once you factor in the site of infection, as various pathogens have different tissue tropism. These individual tropisms change the nature of the immune cell players and the stroma involved. The type of pathogen, viral, fungal, or bacterial, will also dictate which cells respond. Further, the pathogen itself may have acquired virulence factors itself to manipulate the host response. In order to appreciate this complex system, one will need a tool that can capture all parameters. Mass cytometry is such a tool that can not only include many parameters, but can be applied to cellular suspensions or entire tissue sections for imaging. So moving on to some of the publications, the first one is a recent study of COVID-19 patients undergoing treatment for inflammation symptoms caused by the virus shows the power of mass cytometry to visualize both phenotypic and functional changes across the immune system as a whole. This approach enabled systems-level analysis, which overshadowed activation of T and NK cell subsets, which likely contributed to inflammation in the severe cases. Post-transplant T and NK cell subsets were reduced, while regulatory T cell and dendritic cell subsets were increased. It's important to note here that the researchers did not have to guess at which populations to focus on before beginning their study. Mass cytometry provides them the breadth and the depth they needed, especially when they're investigating a new disease such as COVID-19. In another recent study, COVID-19 study, Wang et al. Used mass cytometry for high-dimensional immune profiling of PBMC isolated from patients with different COVID-19 disease severity. Using 35-marker mass cytometry panel, the researchers were able to generate a cluster analysis of CD45 positive single cells, which demonstrate significant differences in healthy donors versus mild, severe, and critical COVID-19 cases. Clusters corresponding to the NK cell population, monocytes, myeloid-derived suppressor cells, and regulatory T cells were generally increased as disease progressed to severe state, but declined in critical disease. Additionally, the regulatory T cells were found to have higher CXCR3, CD28, and TGF-beta expression. Lastly, there was a considerable reduction in all CD8 T cell subsets in all disease groups, corresponding with other reports of lymphopenia in COVID-19. This study from in the meta-archive used a MaxPAR direct immune profiling assay with PD1 and TIM3 added to the profile 50 whole blood samples from COVID-19 patients. The researchers observed lymphopenia hallmarked by decreased CD8 T cell subsets and NK cells as disease severity progressed. Further analysis of NK cells revealed higher activation and TIM3 expression, particularly in critical disease status. The researchers also applied whole blood transcriptomics and cytokine quantification to demonstrate interferon type 1 interferon deficiency in blood as a characteristic COVID-19 severity, and could be used to identify both identify and delineate high-risk populations. Mass cytometry has been used to investigate a variety of infectious diseases. Previous studies had indicated that novel interferon gamma and IL-10 double positive CD4 T cell populations may have a role in severe dengue infection, as IL-10 levels correlate with the disease severity. Using a combination of RNA-seq and a validation of the identified genes by CyTOF, the researchers from the Weisskopf lab at La Jolla Institute for Immunology were able to further characterize these dengue-specific double positive T cells to match a non-cytotoxic TR1 CD4 T cell type that expresses IL-21, IL-22, CD109, and CCR1. Examination of several patients' double positive CD4 T cells showed that there were no differences in the phenotype or function between the varying disease severities. However, the authors could not rule out that elevated levels of the double positive population as a contributing factor to dengue hemorrhagic fever. This study by Cheng et al. Illustrates how mass cytometry was used to determine how multifactorial heterogeneity of HBV-specific CD8 T cells would impact the progression of chronic hepatitis B infection. Using combinatorial tetramer labeling to screen a myriad of CD8 T cell specificities, they were able to also merge this with screening of the 30-marker T cell-centric panel. This enabled them to make both a broad and deep inquiry in antigen-specific CD8 T cells mediated against HBV. They discovered that CD8 T cells directed against HBV core 169 peptide are associated with a long-term memory response, broader trafficking profiles, and polyfunctionality, leading ultimately to viral control. In contrast, low levels of core-specific CD8 T cells and increased exhaustion profile was observed in high viremia. These results generated by this kind of inquiry could inform new treatment studies and determine biomarkers of disease states. This slide here shows a recent report published by the Annals of Internal Medicine. The authors used an 18-marker imaging mass cytometry panel to enable an unbiased exploratory approach to assess immune infiltration in lung tissue from 2 COVID-19 patients, one of who died of cardiac arrest and one from pneumonia complications. Differences were noted in immune patterns of infiltrating CD4 and CD8 T cells, NK cells, and macrophages in a single section of lung tissue from each patient. Recruitment of aberrant CD45 RA positive T cells is an immunologic feature of COVID-19. Once bacterial pneumonia occurs, some phagocytes recruited by CD4 T cells begin to play a significant role in lung injury. In this study, researchers from Yale University aimed to identify mosquito salivary factors that could offer protection against Zika virus infection. They screened the blood of mice bitten by Zika-carrying mosquitoes and found that mosquito salivary protein AgBRI could modulate Zika infection. They also showed that AgBRI antiserum reduced the levels of Zika virus in mice over time, partially protecting them from Zika infection and death. The authors then used imaging mass cytometry to characterize the infiltrating innate immune response to Zika virus infection at the bite site. Differences in levels of CD45 positive, C11b, Ly6G positive neutrophils were observed between mice administered with either naive or anti-AgBRI antiserum, suggesting that AgBRI is involved in the acute inflammation response to Zika virus infection. Together, these findings demonstrate that antibodies against a mosquito protein can offer protection against Zika infection, and that targeting mosquito salivary proteins could be an approach to develop vaccines against Zika and other mosquito-borne diseases. So this section break is just introducing us, moving away from specific focus on infectious disease and now focusing on the actual application of mass cytometry to vaccines. So the power of mass cytometry can also be applied to vaccine research. While the humoral response remains the gold standard for many pathogens, and understanding the humoral response to these pathogens is still incredibly useful to monitor both the innate immune system to early antigen delivery events and downstream T cell mediated immune responses to the vaccine. The graphic shows a simplified example of the immune cascade which occurs upon vaccine delivery by injection, in this particular example. In order to capture a comprehensive view of the response, A proteomic panel with many phenotyping and functional markers is required. Mass cytometry provides this capability. Furthermore, as all vaccines must undergo clinical evaluation, immune monitoring of the response to the vaccine regimen is invaluable. Mass cytometry is a clear, straightforward solution for comprehensive immune monitoring. In this review from the Massachusetts General Hospital, the authors make the case for applying mass cytometry to basic vaccine research. They note that mass cytometry offers a middle ground approach versus lower throughput, high data yield approaches like single cell RNA sequencing or higher throughput, lower parameter flow cytometry. Reeves et al. Also emphasize that mass cytometry enables building of high dimensional panels for assessing numerous phenotypic and functional markers while also being able to screen numerous T cell epitopes via tetramers. Importantly, mass cytometry agents are highly resistant to harsh fixation and permeabilization conditions needed for some types of intracellular staining, as well as the aggressive fixation needed for deactivation mandates of biosafety level 3 practices. There are also published solutions to minimize batch effect, which can potentially confound the results when looking at how the immune response changes over various time points or immunizations. In this publication, Palgan et al. Examined NK cell involvement in the immune response to a common vaccine vector, modified vaccinia virus Ankara, or MVA. Using comprehensive NK cell mass cytometry panel, the author observed significant shifts in the NK cell compartment after prime and boost with MVA. Now, during the boost phase, NK cells were observed to have a distinct functional characteristics as compared to the initial NK response to the prime immunization. The NK response to the boost immunization included increased expression of homing molecules, intercellular adhesion molecules, and cytotoxic capacity. The take-home message of the study is that NK cells, and by extension the innate immune response, can provide considerable insight into vaccine optimization. The final publication to be discussed used mass cytometry to focus on cell-mediated immunity to a vaccine against Salmonella typhi, an intercellular bacterial pathogen. Rudolph et al. Used a high-dimensional mass cytometry panel including 30 unique phenotypic, functional, and proliferation markers to make a broad inquiry of the immune response to the TYR21A typhoid vaccine in both children and adults. Notably, the authors observed that pediatric T cells had more simplistic, limited functional responses than that of adults, who showed a greater multifactorial response. This less robust T cell response observed in children may explain the reduced efficacy of the vaccine in this age group. The study underlines the importance of robust CD8 and CD4 T cell responses in informing vaccine designs in pediatric populations. So that concludes our review of some of the literature focusing on vaccines and infectious disease as mass cytometry is applied to those. I'd like to thank you for your attention, and I would now be happy to answer your mass cytometry and imaging mass cytometry questions. You can enter your question in the question box. All right, one of the questions submitted was, where can we find information about panel kits or either on cytokines or surface markers that you offer for getting started with mass cytometry? So there's a couple ways to do this. One is, I'm going to jump a slide ahead, is that, just to go ahead and show it, is that please take note down of these two particular emails. These are our support portals either for North America or for EU, if you're across the pond in England or Germany or so. But these emails will begin the intake of your inquiry about some of the kits or panels or things you may want to build for your experiment and how you want to integrate mass cytometry into your research program. They will then forward you to a field application scientist such as myself, who can help point you out to some of the products we have or build a custom panel. Otherwise, the Fluidigm website or CyTOF.com will provide a starting point for you to get familiar with some of the panel kits and pre-conjugates we offer to you. Another question is, do you have any pre-optimized panels for mass cytometry? That's a great question, is that a lot of our panel kits are built with other panel kits, or they're built to kind of give you a starting point to conduct an experiment. And we do QC our antibodies to be used at a consistent volume for 3 million cells, but nonetheless, we always encourage you to titrate antibodies to your own purposes. There's one kit that does stand out, and that was in one of the publications that I covered today, was the MaxPar Direct, or the immune profiling system. And this particular panel is 30 antibodies that are lyophilized into a dry pellet at the bottom of the tube, and you only need to rehydrate the lyophilized pellet with either whole blood or PBMC. And to that, what you need for the MaxPar Direct is, it would be 270 microliters of whole blood, and there's a protocol on how to handle that, or 3 million PBMCs resuspended into 270 microliter final volume, and then you will then rehydrate the pellet with that. Another question that was submitted was, can I run intracellular cytokine staining panels on CyTOF? Does it operate any different than ICS staining for flow cytometry? So there is some differences, but really the workflow for mass cytometry ICS is intracellular cytokine staining, that is largely similar to what you would do for flow cytometry. The major difference is at the end of the sample prep or staining procedure is that you'll have to do the intercalation step, which is essentially introducing a reagent that will label double-stranded nucleic acid, and it serves as a single cell discriminator. However, before that, the sample prep, the ex vivo stimulations, and so on, those are largely going to be the same, because you think about this is still cytometry, and you're using similar antibodies just with a different detection and different label on those antibodies. So your handling of those samples to stimulate them with PMA or peptide, as you would want to do in like antigen-specific T cell studies, is going to be the same. And the follow-up question is the analysis. Is that any different? So there is going to be a little difference in the early sample cleanup and gating process to gate out singlets. Is that instead of scatter and forward and side scattered, that you would generally be the first approach with fluorescent cytometry or light-based cytometry, is that you're going to use some Gaussian parameters that our machine puts out, as well as the intercalator that I just mentioned in the last question. Once you have these singlets gated out, then you're looking at phenotypic markers just as you would with fluorescent cytometry. And there's a number of ways to approach data analysis, as many of you know. But manual gating is, of course, welcome. But it turns out to be when you move into the 30, 40 parameter range, it becomes less biased and more convenient, really, to submit your single cells that you gated out manually to a unbiased algorithm of analysis, typically VISNI or dimensionality reductions like that, or cluster analysis like FlowSum, XShift, SPADE, or even Citrus. Another question I got, what are the ideal events to collect for mass cytometry? This is always the answer to this is highly variable, but the best general answer I can give you to event recommendations is you want to identify your rare cell type and work backwards from there. And when you get your rare cell type, so like perhaps it's 0.1% of your overall population, you want to gun for, I'd say ideally 200 events, but probably better to get as much as you can, like 500 events, because if you do use some of the dimensionality reduction algorithms, they do downsample. So it's good to have as much as possible, but you probably want at least somewhere in the hundreds of your rarest event. Okay, waiting for some more questions to come in, but I'll just remind you of FAS Office Hours. If you want to schedule a meeting and just kick the tires in some projects with mass cytometry, just email one of these support portals, and they will forward you to the FAS for your particular region. While we're waiting for more questions to come in, if any more are coming in, I'd also like to remind you or show that there are upcoming sessions, including this particular deck will be repeated on the 18th of September with a different speaker to that time. But the 24th, we'll have a tumor microenvironment analysis vignette, and then on the 7th, we'll do biomarker discovery and then checkpoint inhibitor studies. Furthermore, we have longer session webinars, the top 8 reasons to use mass cytometry, covering a variety of things that make mass cytometry a very valuable tool set for high parameter research or high parameter work, covered by Michelle Poulin, and on the 21st and the 30th. So, and I got another question is, how can I address longitudinal comparisons in mass cytometry? So this is a great question. And the longitudinal comparisons are always a conundrum, and no matter what cytometry workflow you're doing, whether it's flow cytometry or mass cytometry, spectral flow cytometry, is that batch effect exists in every staining procedure. However, mass cytometry has a couple creative tools. Because you have so much parameter space, is that one thing at least between a single run is that you can barcode patients together. With our kits, you can do up to 20 patients, and there are some publications out there doing much more elaborate live barcoding schemes that can combine more than 20 patients into a single stain. Once you barcode, as I said, you combine your patients together or your samples together, and you can then stain them in one tube, thus creating a situation where, well, 20 or more patients, depending on the barcode scheme you're using, has been stained under identical conditions. There are also a variety of publications, as I alluded to in the Reeves review from MGH. There are sources out there to identify batch effect and analyze batch effect. Briefly, one of the most tried and true methods is the idea of taking a known healthy donor or some kind of reference sample that will be present in every sample that you stain downstream, thus giving you a separate set of cells to look and identify if there's any batch effect. Because each sample had the same cell type spiked into it, if there's any differences between one sample versus the other, you could attribute there was some batch effect to this particular sample. The other question that came in was, can you investigate antigen-specific CD4 or CD8 T cells with tetramer and CyTOF? So yes, as a lot of publications from Evan Newells, who's now at Evan Newell, who was at A-STAR in Singapore for a long time, but now is at Fred Hutch, is that you can very much conjugate or build tetramers that have metal labeled tetramers. However, there's one consideration, is that streptavidin in its native form lacks sufficient residues for conjugation, cysteine residues, that is. This creates two solutions to that lack of cysteine residue that you need to address with the conjugation chemistry. Is one, that you can simply use a standard fluorochrome labeled tetramer and then use an anti-fluorochrome metal labeled secondary. And you can simply fold this into your panel. So you do the tetramer stain first. When you conclude a tetramer stain, it's often, at least when I was doing tetramer stains, it was a separate step from my surface stain panel. So you simply throw the secondary against the tetramer into your surface panel for like CD4, CD8, and so on. Two is that as the Newell publications have been, I think the first one of which was in 2013, and since then have been coming in considerably by the year, they take advantage of using a mutant neutravudin, and you can refer to some of the publications or connect with your FAS, and they can get you some of that literature. They use this mutant neutravudin that actually has mutated protein residues or mutated protein that can include free cysteine residues, which is what our conjugation chemistry targets. Okay. I don't see any more questions coming in. I'm just going to go back again, reminding you of the FAS Office Hours for if you have any questions maybe you're being shy about or you want to have a deeper conversation. Again, just reach out to the support portals and they will connect you. And there's the upcoming live sessions. So if there's no more questions, we can hang out for another minute or two in case if anybody thinks of any, but I suppose we will let me thank you for joining today. And again, I really hope to see you in an upcoming webinar session. And I'll just jump back again. Here are some of the webinar sessions. And if you can't remember this, just remember this one thing here, fluidon.com/events. It will direct you to any of these upcoming webinar events. And this will conclude our webinar if there are no further questions. Thank you for attending.